So this video is going to show the detailed process of what happens during both Meiosis 1, which is the first division, and Meiosis 2. So if you look at the very beginning of this cell's cycle, you still have interphase. If I look during interphase, we start off at the cell. It has the nucleus. We've got uncoiled DNA, so it's not visible underneath the microscope. And the most important part here is I'm looking at what's happening during the S phase. And during the S phase is where we have DNA synthesis. That's why it's called the S phase. So our chromosomes are doubling, or they duplicate. So we get the creation of these sister chromatids. Remember, so anytime I see this X shape, we have two sister chromatids. Okay, now remember, they're not condensed yet, so I'm still not going to see them underneath the microscope. So let's look at the very beginning of meiosis. Okay, so I've got Prophase One. So we have some similarities. My nuclear membrane starts to dissolve, shown in the picture here. My centrioles and my spindles start to reform, so that's the same. So here are some differences that happen during Prophase One. So in meiosis, we have our homologous chromosomes, and they start to pair up. And we form something called a tetrad. Okay, tetra means four, so if I would say I have four sister chromatids, I have 1, 2, 3, 4. And when I have these tetrads, or these pairs are forming up, this is when crossing over happens. So if I look in our diagram, that's where literally the chromosomes cross over one another and the genes are switching places. And this is so important because this creates genetic variation. Very, very important. So in Metaphase 1, Metaphase still means middle, but here's a slight difference. My pairs, my homologous chromosomes, my tetrads, they're what going to actually line up in the center of the cell. So I'm still going to have my centrioles on the poles over here. I'm still going to have my spindle fibers, but now they're going to attach. One to this, one to this side, one to this side, and one to this side. So it is the pairs that are lining up in the middle. It is not each chromosome separately. That's what it would have been in mitosis. So in anaphase, it still means apart, but here's the difference. My pairs, or my homologous chromosomes, are going to separate, not my sister chromatids. So I still have my centrioles, and my spindles are going to pull apart the pairs. So I'm going to have this one, and this one over here, and then I got this one, and this one here, and there's going to be some red pieces on this guy, and some red pieces on this one, and on this side I have this one here, and this one like so, and then there's some blue pieces on here. Little guy there, and that one there. So my sister chromatids are not being ripped apart. So if I asked you, do we still have duplicated DNA? We do, because we have our sister chromatids present. But I have no more pairs. My pairs are being separated. Alright, so in Telophase One, you're going to see my nuclear membrane start to come back. So I'm going to draw nuclear membrane coming back in both of the cells. So by the end of cytokinesis, I will have two new, but they're definitely going to be unique genetically, and I'm only going to have one of the chromosomes of the original pair. So I'm going to have my chromosome here. There's this guy. And then this smaller one here, and I had a little bit of red, a little bit of red, 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 and then over here I've got this big one, and then this slightly smaller one, and then a little bit of blue, a little bit of blue on that one, a little blue, a little bit of a blue there. So now here's where we are not done. So now we're going to start the second division, or meiosis 2. So I take these cells here and here, which came from the end of meiosis 1. So these cells came from Telophase 1. So in Prophase 2, you're going to see the nuclear membrane. It's dissolving. So that's my dashed lines. I'm not going to get the tetrads, and I'm not going to get any additional crossing over. Okay, so no tetrads, no additional crossing over. So that I'm not going to see. So that's a difference in prophase 2. So now when I look at metaphase 2, I'm going to see the chromosomes that are left. And remember, I only have 23 chromosomes in each of the cells. The 23 chromosomes will all line up in the middle here. So I don't have my pairs in the middle, but all the chromosomes. So this is kind of similar to mitosis. So now let's look at anaphase 2. Anaphase still means apart, but in anaphase 2, here's the difference. Now my sister chromatids are actually going to separate this time. Remember, in Anaphase 1, my pairs separated. I don't have any more pairs, so now it's my sister chromatids that separate. So I'm going to get one guy that'll get pulled in this direction, and then the other one, I think, had those red pieces on there, so some different DNA. And then I had my smaller blue one down here, and then this one had a little bit of red genes on there. And then over here, the big red one's going this way, the small red one goes here, and then my red one with a little bit of blue, and then another red one with a little bit of blue. And it's going to go that way. So we're almost to the end. Telophase, we're going to see very similar things. So our nuclear membranes start to come back. And so I have to get a nuclear membrane basically around this one, and a nuclear membrane around this one, right? Scratch that. I meant to draw this. A nuclear membrane around this. So that's going to be this cell here. And a nuclear membrane around this side, right? And that's going to eventually turn into here. So in this cell, I'm going to have my big blue chromosome and a small blue chromosome. Then in this one, I'm going to have my big blue one with a little bit of red. I'm going to have a small blue chromosome with a little bit of red. Okay. Then over here on this side, I'm going to have this will be the new nucleus here. This will be another nucleus over here. So that will be, this one's got a big red one here, a small red one with a little bit of blue. Oh, no blues on this one. Mrs. Barnum, what are you doing? Both red. And then this one's got the big red and the small red, and both of those have a little bit of blue on there. So here's what you're going to get at the end of telophase 2 and cytokinesis. So if you look at each of these, I'm going to have genetically unique chromosomes in each of these cells. My pairs are no longer coexistent. I only have 23 chromosomes in each of these cells. I have half of the amount, which is why I classify them as 4 haploid cells. You can either think of I have half of the amount of chromosomes and I don't have my pairs anymore. This is why your sex cells have half the amount of chromosomes, and this is why when those eggs and the sperm are fertilized, that you end up with unique individuals. And it's because of crossing over happened and the fact that you went from a diploid cell to a haploid cell by the end. Just in case you guys see this in a different text, if you ever see 2N, that's referring to a diploid cell, meaning I have both of the pairs. Or if you ever see 1N or just N, that's referring to haploid cells, or meaning I only have one of the pairs. Just in case you see that.